Hi guys, how we doing? How we feeling? Let's put the camera on you if you're not careful. Yeah. Might be your birthday. But I can still be obnoxious. Any day of the week. Okay.
Just kidding.
Okay. I'm writing down names. Okay. So, this is my officially titled quiz number 3 review. It's going to be chapters 6 and 7. I know you guys are probably, if you didn't do so hot or if you did really good on the exam, you want to either keep it up or do better. So this is, I'm trying to prep you guys instead of just asking like, oh, does anybody have any questions and kind of just leading on a tangent. I kind of want to lead and prepare you guys for the quizzes, exams, more so in the future. So let's see. I'm going to start off with the, I'm going to skim through the PowerPoints and I'm going to highlight and I'm going to tell you guys like, this is one you need to know. This is one you need to know. Or like big ideas, concepts that are going to be on the quiz. So let me start with chapter 6. And if people come later, that's fine. But I'll start with chapter 6 right here. Okay, so chapter 6 is all about microbial growth. And remember, that doesn't mean the actual size, but rather the number, right? When we want growth on a plate, we just recently saw that when we did our milk lab, right, that we were looking for growth. And when we saw that growth, it was due to the higher number of colonies we saw. So that's the number of cells that they originated from. Let's see. You guys know this. Living things need energy. How do they get that energy? Metabolism. You guys know that from last exam, right? Right, number rather than size. You know what a colony is. And another little key term that's not really too difficult is just generation time. So that's one cell divides into 2, that's 1 generation time. And then once those 2 divide into, here, let me show you. Because this is a whiteboard. So, oh, that's dead. Cool. I guess this one should work, right? Yeah. So we have 1 cell. The generation time is going to be this right here when it divides into 2 more cells, right? And then this, when it divides again, is another generation time. Okay, just a little visual for the generation time. Okay, speaking of which, binary fission, right? It's not like mitosis. Binary fission is going to be just a lot faster. It's going to be a little bit different because we don't have to replicate all of those complex, like we don't have to replicate the Golgi apparatus. We don't have to replicate the smooth and rough endoplasmic reticulum, right? We don't have to do all of that. Instead, we can very easily just say, okay, I copy my DNA, one's on this one, one's on this end, and then my plasma membrane is going to grow inward and then pinch off, right? It's a lot faster, it's a lot simpler, it's one and done. That's the generation time, right? It's gonna be a little bit more oblong pinch too. And then remember, these are gonna oblong pinch too. Okay. So that would be our binary fission. So yes. So ideally, you know, one of these is going to be, we can see how it can reproduce pretty quickly, right? Okay. And I think you guys can figure this out. It's just constant doubling, right? 1 to 2, 2 to 4, 4 to 8, 8 to 16. Then all of a sudden, when we're at 1,048,576, if we double that again, and make it to our generation number 21, it's going to be 2 million. That's a lot of math in my head. 152. No, it's 100. Yeah, it's 152. Okay. So that would be our division. Just remember that it's basically exponential. It's not like mitosis. So this is kind of the summary of that in a physical form. Know this. This is a key term. This will be on the quiz. So any graph, by the way, any graph. So this is, remember, we have our linear and then our exponential essentially. And this curved, this curved part of the graph, that's going to be time between generations. And that's going to be our bacterial growth. This is a pretty key term. Also know this. So we looked at generation time. This is how one cell multiplies into multiple cells, right? This is the individual cell's growth. So the individual cell will go through a lag phase, a log phase, a stationary phase, and then a death phase. Okay. You can see that there are 2 log phases. One's a log growth, one's a log death or decline. So if you guys follow along with me real quick. So our bacteria has just been born. We have our lag phase here where it doesn't really grow too much. Right? We just had this bacteria start, right? And now we have this exponential growth where not only is the individual cell growing, but it is reproducing. It is doing these generation times. It is producing a lot of bacteria. Then the cell itself that we started with is becoming less and less productive. That's when it kind of hits this stationary area, right? Then eventually the cell will start to expire and come across this death or logarithmic decline, where we will see this cell start dying. And this cell is only a little bit older than this cell. So the cell that we originally had produced this one. This one's gonna die, and then slowly this one's gonna die. This one's gonna die. This one's gonna die, and then suddenly we're left with no more bacteria, right? This is a big staple on the quiz. Know this one. There will be questions on it. Okay. Any questions about any of the generation time, of the phases of bacterial growth, anything like that?
Sorry, I had a question.
Yeah, of course.
Can you explain again what happens in the log phase?
In the log exponential growth or the log death?
The log death.
The log death. Okay, so I'll give you a bit of an idea. Let's pretend like this, where it starts with the lag, is 1, and then just 2, 3, 4, 5, 6 to make it in simpler terms, right? So we start with 1 bacteria here, and it's gonna create its own DNA, grow itself. And then when we go to the log exponential, it's going to reproduce. It's going to do several binary fissions. Its children are going to reproduce. It's going to continue to grow. Then suddenly, let's say that's our colony, right? It has, you know, for simple terms, it has 5 bacteria in this example. These 5 bacteria, they're hanging out and they've reached this plateau. They're not growing anymore and they're not reproducing anymore. Let's say, you know, a number of things can happen, but let's say resources have just run out, right? There's no more food to eat, nothing. So they're going to start to die. So, you know, there's going to be, it doesn't really matter the order, but, you know, as soon as there's no more resources, they're just going to start dying at a logarithmic rate. Does that make sense? Just to define logarithmic, it's just a math term. So it just describes rise over run, how many are decreasing. It's just this rate, right? So if this is our equation here at, you know, let's say log 10, 1, this is what it would be, right? That would be the interception on that graph. It just depends.
Okay. Thank you.
You'll just need to know lag, log exponential growth, has a stationary phase, and then log death. And you'll just need to know kind of what happens in a very basic term of what happens during these phases. Okay. All right. I'm gonna go ahead and move on real quick. Okay. So this is just a few examples of bacteria. You will not need to memorize this. Just understand from this slide that different bacteria have different generation times. Not every single bacteria is built the same. That's all you really need to understand from this slide. Do not memorize more than you need to. Let's see. I think this is kind of like a common sense almost concept. You know, if we figure out how fast microbes grow, we can figure out, you know, the expiration date, right? So if we say once you open a carton of milk that, you know, a typical coliform that's in there is E. coli. So if we say, okay, assuming that it's contaminated with E. coli and we open this, how long until that E. coli produces enough to be pathological, right? Or to spoil the food. We want to know these times so that we can put an expiration date on it, and so that the company that produces the milk doesn't get sued, and that, you know, okay, maybe, you know, I shouldn't drink this because there's probably microbes in it. But we all know the expiration dates are just a bunch of hooey. Okay. Let's see. Know these. So if you guys don't know what the 4 building blocks of any organism are, you can remember it by the acronym CHON, CHON. That's for carbon, hydrogen, oxygen, nitrogen, which are some of the nutritional requirements too. And then we see this oxygen requirement here. And also pH. What's part of pH? The hydrogen, right? So the concentration of hydrogen. So for sure, know this slide, just what these factors are. Remember, the optimal temperature is 37 degrees Celsius, right? That's just so happens, how lucky are we to be our internal temperature too? So bacteria just love our insides. Just peachy. All right. So we got that slide. Some growth factors. This is kind of the same, right? We kind of know from the metabolism point of view that these are essentials, right? We also know that glucose, right? If some bacteria like to ferment glucose, that's going to be a growth factor. Temperature, well, like I said, on the other side, temperature is going to be a growth factor. pH, amino acids, because some bacteria like amino acids over glucose or any other type of 6-carbon sugar. There's a lot of different factors, and it also just depends on the bacteria themselves too. Okay, so let's move on. So for these slides real quick, for proteins, I want you guys to picture cooking an egg, right? An egg is a good source of protein, and when you cook it, you can't uncook it. It won't go back to its original shape because you've denatured the proteins to a point where they cannot go back to that shape. You've broken the bonds and they're denatured and they lose their form. They can never go back. One funny protein that can bounce back is milk, is the milk proteins, right? The proteins in milk, if you were to heat those up, you can, if you put it in the microwave, put it on the stovetop, you can actually have those proteins fold back to their original form if you were to cool them back down to room temperature or the temperature in your fridge. So those are two examples of some proteins that can go back to their original form and some proteins that can't. Most proteins can't. Just think about like cooking food, essentially. But remember, always remember, we learned this in AMP. Structure equals function, right? If we lose the structure in our proteins, they also lose their function, right? Okay. So also Monday, Wednesday, you guys just started your P-Glow lab. Fun. We saw that we had to be careful with our temperatures this whole time. During the entire lab, we had to be very careful with our temperatures. We saw that when the only point where we heated up or gave the heat shock to the bacteria themselves was so that the lipid bilayer would be able to open up for us and that DNA would be able to go in. Otherwise, we also know that DNA is fragile. We need to keep it cold. Okay. So that's another thing that you can visualize in lab and reconstruct it here, reconstitute it here. Okay. I think that's good. Any questions on any of this so far? Okay, let's keep going. So next up, I think you guys understand temperature pretty well. If it's too hot, they're gonna die. If it's too cold, they're not gonna grow. And if it's right in that sweet spot where they absolutely love it, they'll take the opportunity, right? One other thing is that different types of bacteria have different types of optimal growth temperatures. We use 37 as a pretty good internal baseline because a lot of them like to grow there, right? A lot of the mesophiles that we work with, right, the disease-causing ones, that lands right about 37, right? But if we look at thermophiles, right, they're the ones that like a lot of heat. So their optimal temperature is 60 degrees Celsius, which is like, I want to say over like 112 degrees Fahrenheit. So yeah. And then don't even get me started on hyperthermophiles. They are crazy. They're the evil Knievel of the bunch. They are worse than the thermophiles, right? Their optimal growth temperature is like 92 degrees Celsius, which is like, I don't know, like 200 degrees Fahrenheit. That is absurd, right? So this is crazy. So when you look at this graph, the three things that you want to look at is what it is, what's the lowest temperature it'll go, what's the highest temperature it'll go, and what is its sweet spot? What is it like the most, right? These ones that are circled right here, no for sure. This over here, why it's circled, is just so you can see it, right? The minimum, maximum, and the optimal. But you want to know your mesophiles, minimum, maximum, and optimum. And then you want to know your psychrotrophs, minimum, maximum, and optimal. And just know what they are. That's the only takeaway, is those two humps and the information around that. And if you download the slides, sometimes you can get a little extra tidbits of information on the bottom. So there's that too. So just keep those in mind. So here, what do we see, right? We see a lot of ice, and all of a sudden we have this bacteria growing on top of the ice. Wait, I thought it was 37 degrees Celsius. Well, this one likes really low temperatures. We could probably say that it's one of these, the psychrophiles, right? It likes really low temperatures. This, on the other hand, if you've ever been to Yellowstone, I personally have, and I would recommend you guys go, but these baths are always boiling. These are where the thermophiles like to live. And they have special membranes, right? So those are just a couple of examples, right? And you see how we go more in depth with the psychotrophs. And we want to also understand food spoilage, right? What's the bacteria in rice that can spoil very easily and give you very bad food poisoning? You can't be serious. You didn't like what I did there. Okay, enough of the puns. But it's Be Serious, right? Be Serious is our famous one for rice poisoning. Let's see. Again, this is just most, right? We went from temperature, now we're at pH. So 6.5 to 7.5 is a sweet range for most bacteria. Molds and yeast, 5 and 6. Acetophiles, though, we see the word acid and we see the word phile. So what do we think? They love acid, right? So they're going to have the lower end of they're going to be below basic all the time. Okay. I think that all you really need to know here is what is a neutrophile, what is an acetophile, and that's essentially it. Also know Helicobacter pylori, that is found in our stomach. And what do we have in our stomach? Stomach acid. Acid, stomach, bacteria. Is it a neutrophile or acetophile? Right? But yeah, so just think of things like that. Let's see. Yeah. That's all you'll need to know for pH, is just what are the ranges for different things like this slide, and then define neutrophile, acetophile, and give an example of probably each. Okay. Gross, I know. But remember that as we get older, you know, our immune system can't really hold off our own microbacteria anymore, right? So we need to also understand that, you know, stomach ulcers exist. So our own Helicobacter pylori can cause stomach ulcers. If you're really stressed out, or if your diet's not right, and there's, you know, if you have too much acid in your stomach, then you know, like acid reflux. If you get acid reflux a lot, if you get heartburn, if that's too much acid in your stomach, too much Helicobacter pylori, you need to do something about it, or else you could get stomach ulcers. And that gives you a risk for gastric cancer. No, the stomach ulcer, this definitely. Also, isn't that crazy how they fed a tube all the way down to the sphincter in your stomach to see that little ulcer? It's gross, but it's kind of cool. Le cholera. So know a little bit about cholera, right? It's a strange one because we learned about acetophiles. Now we need to know about alkali files. I can't speak today, I'm sorry. But basically the alkaline version, right? It's the basic alkaline. They like a more basic pH to grow in, right? Also know that they like shellfish for some reason. I don't know. But shellfish, most of them live in fresh water. So just know kind of those factors for this disease. You guys probably know hypotonic, hypertonic, took chemistry. You guys understand this idea right here. Right, this was part of, I think, correct me if I'm wrong, but Bio 33, where if we have a nice equilibrium, right, it's isotonic. Our red blood cell is going to stay a nice natural shape, right? But if it's hypotonic, where there's too much pressure on the inside and there's not enough pressure on the outside, it's going to swell and burst, right? But if it's hypertonic, where there's too much pressure on the outside and not enough on the inside to keep its structure, it's going to shrivel, right? And we don't want the last two, so we like isotonic solutions. But again, if it's a certain osmotic pressure, maybe the bacteria can't reproduce. Maybe the bacteria doesn't want to produce if it's not a specific osmotic pressure, right? So know probably this out of the slide. Also, you know, what makes bacteria happy? A nice temperature, nice warm conditions, nice moist environment, and a lot of food, right? And a nice little even environment. Just think about getting cuddled up on bed and you have your little cup of coffee, right? So cells need a lot of different things. That's the gist of it. So can you guys tell me, I'm going to see. Basically, if you guys want extra practice, I believe the answers are in here, or she has gone over this with you already. But do this. Do not try to just jump ahead and be like, okay, I'm just going to write it out. Try it and test your understanding, because I guarantee that you'll think you have it and then you don't, and then you have to go back and be like, okay, yes, now I understand this, and it's a learning moment. Being wrong is a learning moment, I promise you. I hate to call it being wrong. It's just, you know, more the practice makes perfect, right? But definitely do this. Check your answers with the lecture, and it'll be amazing practice. And if you have passed chemistry homework with this, maybe look back at it if you're a klepto like me and keep everything and steal extra worksheet pages and stuff like that. Okay, so we talked about osmotic pressure. It's similar to the red blood cell, right? So hyperosmotic shock, right? If the solution is hypertonic, it's going to die of hyperosmotic shock because it's going to desiccate. It's going to shrivel, right? Let's see. Man, we love this one. Okay. what is our favorite halophile that we've been testing for on MSA plates? You guys know it, I promise you do. If you guys want to type it, that's cool too. Oh, I got one. Yes. S. epidermidis is one. It's okay. I don't care about spelling. You're fine on spelling. But right, it's they love salt. Halophile loves salt, right? We have the obligate versus facultative, right? Obligate, love it. I can't live without it. But facultative is like, I can tolerate it, I guess. You know? And look at this down here, if it'll let me do it. These are the 3 main concepts you need to know from this slide. It'll probably be on a quiz or test in the future. Okay, so I'm going to finish this up and then we're going to take a 5-minute break and then we'll come back and I'll do 7. But really quickly, we know hypertonic, we know this, right? This is the same thing with the plasmolysis. If it's in a hypertonic environment. Let's see. Oh, a nice carbon food source, right? Our 6-carbon sugar, that's a carbon food source. It could be glucose, lactose, galactose, any of those things. If it's a 6-carbon sugar, we're good. That's our carbon growth requirement. Here, look, it's Chan, but there's 2 words mixed up, right? 2 letters mixed up. It's also a band, if you guys listen to them. But this is a good Tron, you know, I guess it rhymes. If you guys like that movie, I don't know. I've personally never seen it, but I digress. So these are some of our other chemical requirements, right? These are some more, but these are just other elements, right? And then trace elements. But these small amounts, this large amounts, right? This is 95%. This is the other 5%, right? Big gap. Oh, and look, this is all you need to know from this slide. Okay. Nitrogen, yes, amino acids. So nitrogen and amino acids, they love each other. They're just, can't live without them, right? Okay, that's basically all you need to know there. Sulfur, amino acids, thiamine, biotin. You guys know this, biotin, nails, hair. That's about, I think that's it. Eyes, I think, but don't quote me on that. At least in your nails and hair. So just know that you need sulfur for those things. You guys probably know the phospholipid, the ATP and ADP, and the nucleotide structures, right? If not, review this, draw it out. It's great practice, okay? Also, you guys covered enzymes in the last exam. So what the point of this slide is saying is that during some metabolic processes, you need the cofactors, right? And the cofactors in bacteria can be the trace elements. Usually the trace elements are cofactors. That's just the basic of this slide, right? So if we don't have these cofactors, right, we don't have enough magnesium in the bacteria, or if it's somehow lacking the production or absorption of those trace elements, we don't have a cofactor, we don't have an enzymatic reaction. Okay. You guys saw about this with your F tubes, right? Just if you can read it, define it, you're good for the exam, okay? look, these are just the words you need to know and be able to define, right? And this is going to be our broth that we saw, right? So if you can go through these and cover up the bottom and tell me what they are, perfect. You are prepped for the quiz. Also, make sure that you look at this, right? So you'll need to know the 4 toxic forms of oxygen, right? And know that we're not just defenseless with this, right? We have certain enzymes, right? It ends with an ASE. We have certain enzymes that can break down the toxic forms of oxygen so that they can live in our bodies and be fine, right? Also, this superoxide dismutase, you'll need to know it, memorize this equation, and you guys will see it in lab, okay? This is, so look at this one, right? We have catalase here. So if we start with, if we start with hydrogen peroxide, right, it's going to turn into water and oxygen. So if we have if we put hydrogen peroxide onto a organism that has catalase, it's going to break that down into water and oxygen. That's why we see these bubbles. That's why we see the production of water, right? So this is our catalase test, is by adding hydrogen peroxide. You guys will do this at another time, okay? Are there any questions on chapter 6? I'm gonna stop the share momentarily. And I have a few more that show. Okay. So if I don't have any questions, or if you'd like to type a question, I know sometimes unmuting is a little tricky. But if you have a question that you don't feel comfortable speaking out loud, remember the recordings, I don't post the chats, so I just read a question and it could come from anybody. So if you have a question, I'll answer it for you. And if not, I'm gonna go ahead and give you guys a break. It's 5:37. I'm gonna give you an extra minute. It's 5:38 now. I'm gonna give you an extra minute anyways. So come back at 5:44. 5:44, and we will go ahead and wrap up chapter 7. This is the other half of the quiz, okay? So come back and we'll do chapter 7, and then let's see. Yes. So we'll do chapter 7, and I will be able to wish you guys luck on the quiz. Just so you guys know, Wednesday, I will have another review session, 7 p.m. And that one will be more about the Piccolo lab and some math with the milk lab for like dilution and dilution factors. And then I'll probably continue that on Thursday at 11:30 in the morning too. But yeah, you guys can go ahead.